And basic the idea is our genetic material is DNA. So through replication, it can replicate itself. And to make the functional molecules, it will produce the RNA by transcription. And that is the topic we are going to talk about today. And then after that, the RNA will be translated into the proteins. Of course, we know that there are many more different kinds of RNA now. So here I show you the figures of where is the location of the replication and transcription. Basically, replication and transcription occurs in the nucleus of the cell, and this is part of the nucleus of the cell, and the translation occurs in the cytoplasm of the cells. Before going to talk about transcription, I want to make some comparison between replication and transcription. So both of them have some common features, and of course, they have some different features as well. They both can synthesize a new strand of a nucleic acid. For replication, it will make a DNA strand, and for transcription, it will make a RNA strand. And for the most feature of the transcription, that it composed of the ribonucleotides, not the deoxyribonucleotide in the DNA. And of course, the base was different too. And in DNA, there will be A, T, C, and G. And in RNA, there will be A, U, C, and G. So the difference is T in DNA and U in RNA. So for transcription, this is the RNA polymerase responsible for making the RNA. And for RNA polymerase, it does not need the primer to initiate the transcription. However, in DNA polymerase, it needs the primer to start synthesize the DNA. And in comparison to DNA polymerase, RNA polymerase is less accurate than replication. It's about 1,000-fold difference. And the most important part of transcription, it can be respond to different stimuli, and it will have different levels of transcriptions. So here shows you the basic structure of the transcription. In the middle, there is a transcription bubble. So in this oval structure indicates the RNA polymerase. And in the middle of it, it is the 2 DNA strand, which shows the blue helicase here. And these 2 DNA strands are separate from each other, which means they are melted. And the red line here indicates the transcribed RNA. So you can see that the transcribed RNA in the transcription bubble is hybridized with the one strand of the DNA. And this strand of the DNA is called the template strand DNA. And it's also called non-coding strand and also antisense strand, because it has the opposite base composition of the transcribed RNA, therefore it's called the non-coding strand. And the other strand of the DNA is called the non-template strand. And since it has almost the same sequence as the coding RNA, therefore it's called the coding strand. Now we are going to talk about the RNA polymerase. Basically, it needs the DNA as the template to do the synthesis. And for prokaryotic RNA polymerase, there is only one kind of enzyme. Whereas in eukaryotic, there are several different RNA polymerase. I will talk about more detail later. And for both prokaryotic and eukaryotic RNA polymerase, they all consist of several subunits. And their molecular weight of the whole complex very huge. For example, for the prokaryotic RNA polymerase, it's about 480 kilodalton. It's very huge. And for eukaryotic RNA polymerase, there are three kinds. Majorly, there are three kinds of RNA polymerase, and each one of RNA polymerase is responsible for making different kinds of RNA. So I already talked about that. No matter is prokaryotic or eukaryotic RNA polymerase, it consists of several different subunits. Here is the table shows different subunits of the RNA polymerase. Here is the prokaryotic RNA polymerase, and this is the eukaryotic polymerase. So you can see there are lots of them. You don't really need to remember the name of each one. So basically, you just need to remember it's composed of multi-subunits. And for the eukaryotic RNA polymerase, there are basically three kinds of RNA polymerase. So in short, RNAP means RNA polymerase 1 is responsible for making large ribosomal RNA. And RNAP2, which indicates the RNA polymerase 2, responsible for making messenger RNA and most small nuclear RNA and microRNAs. And for the type 3 of RNA polymerase, is responsible for making transferrin RNA, 5S ribosomal RNA, and other small RNAs. And especially in plants, there are actually two other kinds of RNA polymerase, 4 and 5. They are making small interferon RNAs. So you already have some basic ideas for transcription. You need the RNA polymerase to synthesize RNA, and then you need to separate two strands of the DNA. One strand is template strand DNA, and the other one is the non-template strand DNA. So I'm going to talk about more details now. Basically, there is three different topics I'm going to talk about. First, I'm going to talk about more details in prokaryotics, and then later in eukaryotics. And for the last part, I will going to talk about what's the importance of transcription in cancer. So I already talked about there is only one kind of RNA polymerase in prokaryotics. But we can take a careful look. They actually compose of 5 different subunits, 2 alpha subunits, and 1 beta and 1 beta prime, and the omega, sorry, is behind the figure. And the 5 subunit consists of the core enzyme. And then the core enzyme is already have capability to synthesize the RNA. With the addition of extra factor, which is called the sigma factor, it completes and then it becomes the holo enzyme. So what does the holo enzyme does? It can initiate the transcription from the promoter. Whereas the core enzyme, it also can synthesize RNA, but it can initiate transcription at any place, at any sequence. So this is the sigma factor do. The function of the sigma factor is to recognize the promoter, and it can initiate for the transcription. So the sigma factor is kind of guided RNA polymerase to be recruited to the promoter, which is really important. So this just shows again the same idea that with the addition of the sigma factor, it becomes the holoenzyme of the RNA polymerase. Without it, it's just the core enzyme. And the core enzyme already have the capability to synthesize the RNA, and it can elongate the transcript. But it cannot initiate the transcription at the promoters. So there are three phases of the transcriptions. So the first phase is the initiation phase. The initiate phase, the RNA polymerase need to be recruited to the promoter and bind to the DNA template, start to separate two DNA strands, and then start to synthesize a stretch of RNA. And then the second phase is to elongation. The RNA polymerase will synthesize a lot of, it continues synthesize the RNA until the end of the gene. But how does it know where or when to stop? And that enters to the third phase of the transcription, is the termination. So when you encounter some signal, and then the RNA polymerase needs to leave the DNA template and release the transcribing the RNA. So I will go into more details for each phase of transcription. First, I will take a look of the promoter. So promoter is the DNA sequence that initially binds by the RNA polymerase, and that is the very important DNA element. And after the RNA polymerase binds to the promoter, and it will start to separate the two DNA strands around the promoter, and it forms the transcription bubble. The RNA synthesis occurs in a 5' to 3' direction. So we can see that although there is no showing the figure of the sigma factor, actually is the sigma factor responsible for recruit the RNA polymerase be located to the promoter. So after the RNA polymerase binds to the promoter, it forms this DNA enzyme complex. At this stage, because the two DNA strands still hybridize with each other, so it's called the closed complex. So what's the feature of the prokaryotic promoter? There are actually three features. One is the -35 region. So what is the -35 region means? We have to take a look of this +1. The +1 means the transcription start sign. And usually we will put the +1 like here, and then draw an arrow and indicating the transcription orientation. So the -35 region, meaning the region actually is about 35 base pair upstream of the transcription start sign. So you can see in this region, it consists of 6 base pair nucleotides. And the second feature of the promoter is this -10 region. And you can see this is the 10 base pair upstream of the transcription start site. It also consists of 6 base pair of nucleic acid. So the third feature of the promoter is the space between the -35 and -10 region, which you can show is in a range of 17 to 19 base pair. In these figures, I only show the consensus sequence of the -35 region and -10 region. you can see that in the -10 region, it actually is rich in AT pairing. And the most conserved distance between -35 and -10 region is 17 base pair. It's not every gene has the promoter sequence, just like the consensus sequence. But when it does, it have a very strong transcription going on. So what does this strong mean? It means that if it's really strong, then there will be many transcripts can be produced in a given time. So there will be a lot of transcripts. So if the gene has a promoter, the sequence is less like the consensus sequence. The transcription will be really weak. So I already talked about is the sigma factor responsible for recognizing the promoter regions. So I'm going to show you the diagram of that. This part of the line here indicates the sigma factor, showing the primary structure. You can see this is the C terminal of the sigma factor, and this is the N terminal part of the sigma factor. You can dissect the sigma factor into 4 different regions, which is 1, 2, 3, and 4. And of course, you can make it subdivide it into very small region like 4.1 and 4.2. And you can see that the sigma factor used the section 4 binds to the -35 region of the promoter. And it used the section 2 and binds to the -10 region. And this is the place where the 2 DNA strands started to separate to each other. So for the initiation phase, after RNA polymerase binds to the promoter, it forms the closed complex. And for the next stage, it will start to open or separate the 2 DNA strands around the promoter. Usually is in a distance of -11 to +3. And you can see in the figure here, this part, the 2 DNA strands are separate. And then because 2 DNA strands are separated, at this stage, is called the open complex. After that, the RNA polymerase start to synthesize the RNA. So you can see a small stretch of RNA, which color in green. And you can see that at this stage, there are ternary complex composed of DNA, RNA, and the enzyme. And at this stage, the transcription bubble forms. So once it starts to synthesize a stretch of RNAs, usually it needs to over about like 9 base nucleotides long of RNA, and then it can kind of give RNA polymerase a single signal, and then it will push the RNA polymerase to leave the promoter, which means indicates the promoter escape. And this is the time for the RNA polymerase enter to the second phase of transcription, which is the elongation phase. So I'm going to show you again about the transcription bubble. At this time, I want to emphasize about the DNA duplex in front and behind the transcription bubble. So you already know this oval structure indicates the RNA polymerase. And the 2 DNA strands, which one is in blue and the other one is in red, they are separated. And the transcribed RNA is in green. And in front of this transcription bubble, you can see that the DNA strand have to unwind because the RNA polymerase will keep moving to your right side. And then the DNA duplex behind the RNA polymerase need to reanneal back to each other. So this transcription is very dynamic process. For the elongation phase of the transcription, it will keep making and synthesize the RNA. So you will keep doing the DNA unwinding, DNA reannealing. And at any time, the RNA-DNA hybrids only about 8 to 9 base pair. The rest of transcribed RNA will be exit from the exit pore of the RNA polymerase and hanging as a single-stranded. The RNA polymerase possess a processive mode, indicates once it starts to synthesize RNA, it will keep synthesize a stretch of RNA, and after it finish, it will fall off. And of course, it has the proofreading function. So at any time, it will retract back a little bit of several nucleotides and correct it, and then keep doing the synthesis again. So for the last phase of the transcription is the termination. When does the RNA polymerase know to leave the DNA template and releasing the whole transcribed RNA? So it has to have some signal to tell RNA polymerase to stop, which is called terminators. The terminators is the sequence that trigger the elongation polymerase to dissociate from the DNA. There are already two different mechanisms of terminators. One is role dependent and the other one is role independent. So what is the role? Role actually is a single-strand RNA binding protein. And usually it functions as a hexamer. So it consists of 6 identical subunits. And when the role binding to the RNA, it was use the energy from the ATP hydrolysis, and then it will rest the RNA from the RNA polymerase. So where does it bind? It actually binds to the ROT, meaning role utilization RNA size. And importantly, role does not bind the translating RNA. In prokaryotics, we already know that at most of the time, the transcription and translation will occur at the same time. So if that happens, rho does not bind the translating RNA. So this is the figure showing how does the rho doing for terminating the transcription. However, I have to point out that there are still many detailed mechanisms still under debating, still researchers still doing their investigation to know the detailed mechanism. So in this figure showing that the rho protein is a hexamer, it will bind to the RAS side. And the RAS side, what's the feature of the RAS side? It actually is rich in C residue, cytidine residue, and usually it will contain about 14 nucleotides of single-stranded, which is really unusual for the single-strand RNA, because for the single-strand RNA is naturally it will form the secondary structure. So The rho factor is a hexamer, is a ATPase and a helicase. So you will use the ATP and then as an energy to unwind the RNA from the RNA polymerase. So another mechanism for the termination of transcription is the rho-independent terminator. So in this case, we don't need the rho protein. And the only features for the terminators is inside the sequence itself. So you can see that this is the DNA sequence, and this is the transcribed RNA. In the DNA sequence, actually it contains the inverted repeat. So when you transcribe the RNA, it can form this stem loop structure, also called a hairpin structure. And then after followed by the inverted repeat, there will be a stretch of AT pairing. It's about 8 nucleotides long. And AU pairing is the weakest base pairing among all of the base pairing. So you can see that once this kind of RNA forms, the stem loop structure forms and it will store the RNA polymerase. And then again, the detailed mechanism is still unknown, but people think that maybe it will cause the conformation change and then will let the RNA polymerase leave from the DNA template. So I already finished up the transcription in prokaryotics. I'm going to talk about more details in eukaryotics. Basically, the basic idea is the same. Just in eukaryotics, they are more complicated. Like I mentioned earlier, there are 3 different kinds of RNA polymers in eukaryotics. So the RNA polymerase 1, 2, and 3. And for the eukaryotics, there are some additional factors will help transcription to occur. And this is called the general transcription factors, in short, TF. So for each different RNA polymerase, 1, 2, and 3, there are transcription factors 1, 2, and 3. So the function is similar as sigma factors. As you can recall that the sigma factor is help RNA polymerase to be recruited to the promoter. And that's what some function of the general transcription factors do. So here it shows you the core promoter of the RNA polymerase 2 that you can see is more complicated than the prokaryotic promoters. You can see that this is the TATA box, TATA, because it consists of the TATA sequence. And the element upstream of it is called BRE, is the B recognition element. And downstream of that is, in short, INR, is initiator element. And for the more downstream part, element is DCE and DPE. So the DCE is downstream core element, and DPE is downstream promoter element. So you already think it's very complicated, but in different genes, actually, you don't really need every element. So for example, some genes, you have the BRE TATA box, but you don't have INR and you have DCE. So here we only shows the all core element that it can contain. So you have to remember that this core element, this core promoter is only for transcription initiation by the RNA polymerase II for in vitro. For in vivo, they are more complicated. So you can see that for initiation of transcription, they are more stage. You can see that here the TF2D is the transcription factor 2 called D, and the TF2D contains the TBP. The TBP is called TATA binding protein, and it also contains TAPS. It's called TBP associated factors. So even the TBP actually consists of multi-subunits. The TF2D, this big complex, is responsible for recognizing the TATA element. So once the TF2D recognizes the TATA box element and it binds to it, and then you will recruit the other two transcription factors, TF2A and TF2B. And the TF2B will bind into the BRE element because the BRE is the responding element. Sorry, and then after that, you will recruit the RNA polymerase. The TF2F actually will bind to the RNA polymerase and then be recruited to the DNA. And then after that, the TF2E and H will also bind to this whole complex. And this is the pre-initiation complex. And you will start to separate two strands of the DNA. So you already can see that they have more different transcription factors and more proteins needs to bind with the RNA polymerase to initiation for the transcription. And for the promoter escape, it also is more complicated than in prokaryotic system. So in prokaryotic system, the RNA polymerase need to synthesize a stretch of RNA, and then it needs to over like 9 base pair, 9 nucleotides of RNA. But in eukaryotic transcription, it also needs to transcribe over 9 nucleotides of RNA, but it also need another important feature. You can see that RNA polymerase, actually there is a tail here I didn't mention earlier. This is the very special feature of RNA polymerase in eukaryotics. This is called the CTD tail, the C-terminal domain tail. The CTD tail actually contains a heptapeptide repeat. So the sequence is showing here is composed of tyrosine, serine, purine, threonine, serine, purine, serine. So it will these 7 amino acid repeat several times, and in different species, the repeat time is different. For example, in yeast, this 7 peptide repeats about 26 times, but in human, this repeats about 52 times. So it seems that more complex organism, it contains much more repeats of this heptapeptide. So after it synthesizes a stretch of RNA, and then the CTD tail, actually you can see is showing the P here, indicates it's phosphorated. So before, for the initiation of transcription, the CTD tail is unphosphorated. Once it starts to ready for leave the promoter, the CTD tail will be phosphorylated. And this is the feature for the promoter escape. So I already talked about there is a core promoter element, but actually there are more complicated systems in eukaryotics. So I'm going to show you more about that. So first we look at this figure here. This is the Tata box, and actually there will be UAS, upstream activator signals, and upstream of that is the silencer. The silencer is the repressor. Is the some transcription factors actually will bind to the silencer. And what does it do? It actually, after it binds to the silencer, it will terminate the transcription. So basically, it will be repressor to stop the transcription. And in other genes, or in many genes actually, it will have multiple different regulation of the transcription other than the core element we already talked about. So you can see this is the core promoter region, and this is the silencer, which will be bound by the repressor. Also, you can see there will be an enhancer over here, So enhancer, it can be in a distance over like 1 mega base pair. It's kind of far away, but it still can have effect on the transcription. So once the enhancer will be bound by the activator, it will activate the transcriptions. So you can see that different regulation can occur on the same gene and affect its transcription. So from previous slide, I already talked about there will be several different control elements to control the transcription, but we only talk about is the naked DNA, meaning that is no nucleosome was present. But in the reality, in vivo, the DNA was wrapped with the nucleosome and is in the chromatin structure. But how does when the gene is going to be transcribed? So what this nucleosome does, we have to kind of push it away, or to let the transcription can goes on. So we need other factors to modify or to remodel the chromatin. So this is called the chromatin remodeler, or like a chromatin modifier is called this one in the case is the HAT, is the histone acetyltransferase. It can kind of doing the post-translational modification on the histone, and then it will activate or deactivate the transcription. Also, there are another big complex called mediator. The mediator complex is actually consists of 20-ish different subunits. It is co-activators. The function of the mediator is kind of transmit the signal from the activators and then to the RNA polymerase. So it's kind of like a bridge. So you already know that there are several different regulation can affect the transcription. So again, this just showing that for just any kind of gene that there, this is the transcription start site, and then this is the orientation of the transcription. And you can see this is the core element, and then this is the mediator and the chromatin modifier gene, and also there will be enhancer. Many different proteins, many different enzymes, or many different DNA elements can affect how strong the promoter are, and then how many transcript it can produce. So after it can be initiated, and of course it will go into the next stage for the elongation. And for the elongation, it also needs more proteins to stimulate this phase. So, and another important feature of eukaryotic transcription is the RNA needs to be processed further. So there are more RNA processing factors, including the capping enzyme and the splicing that I'm not going to talk about today. And also we have to adding the poly A tail at the end of the messenger RNA. So what is the capping do? The capping actually, it would doing this, use this methyl guanine to use a very unusual 5' to 5' linkage to the start of our messenger RNA for this, and this is called the capping. The capping will protect the RNA from degradation. Also, it will increase the translational efficiency. And the splicing is going to splice out the introns. That I'm not going to talk about today. And the final part is going to adding the poly A tail at the 3' end of the RNA. So for the polyadenylation, there are two important factors for doing this. So the first one is the CPSF, is called the cleavage and polyadenylation specificity factor. And the other one is CSTF, cleavage stimulation factor. So initially, these two factors are bound to the CDT tail of the RNA polymerase. Once it found that there is the poly-A signal sequence in the RNA, these two factors will jump to the RNA and then doing the cleavage. And it will release the RNA from the RNA polymerase. And then later the CSTF will leave and CPSF still remain bound to the RNA. And then it will recruit another protein, is called poly-A polymerase, in short term PAP. So the PAP will bind to the 3' end of the messenger RNA. And then it will, without any DNA template, it just add extra stretch of the A. And then you can see this poly A tail is about 200 nucleotides long. So for the termination of the eukaryotic transcription, the current accepted model is called torpedo model. So basically, right now people think once the, although we already have the messenger RNA is with the addition of the poly A tail, the RNA polymerase still remain on the DNA template and still doing the transcription. It does not know when to stop. So when does it stop? It need another factors. It's called the RET1 in yeast, and in human it's called XRN2. And you can see it originally is on the RNA polymerase. So, so you can see that after this messenger RNA is cleaved, and then you can see with the addition of the poly A tail, there is still a short stretch of RNA bind with the RNA polymerase, and then this XRN2 is found. Okay, this RNA is uncapped. Because the original messenger RNA is 5' capped. So XRN2 found this is the uncapped RNA. So it will start to do the degradation. It will chew the remaining RNA. And then after it degrades all of the RNA polymerase will leave. So you can see that the elongation, termination of transcription, and RNA polymerase RNA processing are interconnected to each other. So this is the summary of the whole topic I'm talking about today. So I did talk about the most important for the transcription is the RNA polymerase. We need some factors to guide RNA polymerase to be recruited to the promoter and start to do the initiation of transcription, elongation, and termination. Of course, in different phases, we all need different factors to help the process. And for the termination in prokaryotics, there are two known mechanisms for role independent and role dependent. And in eukaryotics, they are more complicated. Other than the promoter, the RNA polymerase, we need general transcription factors, and also we need activators and repressor, and also we need to modify the chromatin and the mediators. So it will be more complicated in the eukaryotic systems. So for the final part, I will going to very quickly just going by. This was mentioned about the hallmarks of the cancer. So for the cancer cell, of course, we already know that they will behave very different from the normal cells. So this paper showing there will be 6 hallmarks of the cancer. Of course, the author actually, they in this 2011 paper, they mentioned 2 additional hallmarks I'm not going to talk about today. So the most important for this part is how can we keep the cancer cell immortal. So you can see that this is very complicated. So the cancer cell will proliferate more than the normal cell, and it will move faster, or it can invade, or it can metastasis, or also it can immortal. So all of the things, how can cancer cell do these kind of things? Because it can have dysregulation of the gene expression. So that's lead to the transcription part. So I'm going to focus just how do cancer cell immortal. For the normal cell, because we have, this is just showing the chromosome. At the end of the chromosome, we have the telomere. In the normal cells, our telomere will become shorter and shorter, so that's why we are not immortal. And for genetic recombination experiment, we can introduce some extra antigen or some mutations that keep the cells they can proliferate more, and then they can become immortal. So to let the normal cell become immortal is to introduce the telomeres. The telomeres will increase our telomere, the length of our telomere, and then that will make the cancer cell immortal. And that will be related to the paper that students will present today. So that's the end of my talk. Thank you very much.
So any questions? So no questions? Very clear? So probably the professor should ask questions. So any questions, Japan side? Okay, so if there's no any questions, so we probably take a 5 minutes break and the student can prepare a discussion. Okay, so let's take 5 minutes break. So okay? All right, so we have,
Yes.
okay, so we have 5 minutes break. Thank you very much
Thank
for the
you.
professor. Hello, shall we get started?
This is Kelsad. We are all ready.
Okay.
Yes, school side also.
Okay, thank you. So we are moved to the student presentation. So today they are going to present a paper from science. So I guess the Professor Huang already mentioned about the telomere. Okay, so those are third promoter in cancer. So let's welcome student and then go ahead.
Hello, everyone. I'm Wang Yushen for our presentation. First of all, I'm going to introduce my team members. Over here is Qi Zhang, and next to him is Zheng Ting. The next one is Di Xu, and the last one is Shou Ting. So the topic of today's presentation is the transcription factor gap selectively binds and activates the mutant TERT promoter in cancer. According to the American Cancer Society, 1 out of every 2 men and 1 out of every 3 women will be diagnosed with cancer. Moreover, it's necessary to know that cancer is already the leading cause of death in Taiwan and Japan. And based on the fact sheet from WHO that nearly 1 in 6 deaths is due to cancer globally. But despite those huge numbers, most people don't really know what cancer is and how it happens. At the simplest level, cancer or cancer cells are cells that have lost the ability to follow the normal control that the body exerts on all cells. Every cancer starts when one cell in the body begins to divide uncontrollably. The rapidly dividing cell pushes aside the healthy cells around them. They form a small tumor, and they start to invade into adjacent organs. To have the energy to keep growing, the cancer needs a blood supply. To get that supply, the cancer sends out chemical signals that draw blood vessels to it. Unfortunately, these vessels allow cancer cells to spread to other parts of the body, and that can be life-threatening. Once the cancer has spread, it's harder to cure. But what makes a cell turn cancerous and divide uncontrollably? Cells divide when their genes tell them to, but if a gene has a mutation, it can tell a cell to divide when there's no reason for it to divide. Recent research has discovered that mutation in the TERT promoter of the TERT gene are the most common genetic alteration in multiple cancer types. During development, the TERT gene silencing in most somatic cells. And as a result, telomere shortening during chromosome replication. When telomere become too short, the DNA damage response will be activated. This response can induce apoptosis or senescence. However, since the mutation happened in TERT promoter, it results in a barren expression of TERT. This event allows the chromosome with unstable genome to survive and become cancer cells. But what is telomere and why telomere shortening during chromosome replication? A telomere is a region of repetitive nucleotide sequences located at each end of a chromosome. For vertebrates, the sequence of nucleotides in telomeres is TTAGGG, and these repetitive nucleotides are essential for genomic stability. The addition of these nucleotides is catalyzed by TERT, TERT is a catalytic subunit of enzyme telomerase, which together with telomerase RNA component comprises the telomerase. Telomeres protect the ends of chromosome by forming T-loop, which avoid chromosomes sticking to neighboring chromosomes. Not only that, telomeres also protect the chromosome from DNA damage during chromosome replications. During chromosome replication, DNA is always synthesized in the 5' to 3' direction. As a consequence, the lagging strand is synthesized in short, separated segments. These segments called Okazaki fragments. Their synthesis requires RNA primers attaching a head on the lagging strand. Then, RNA primers are removed and replaced with DNA. However, at the end of chromosome, there is no 3' hydroxyl group available to prime DNA synthesis. Because of this inability to replicate the ends, chromosome would progressively shorten during each replication cycle. An end replication problem is solved by the telomeres. The repetitive nucleotide sequences of telomeres protect the chromosome from DNA damage during chromosome replication. For these reasons, telomeres are essential for genomic stability. During development, telomerase is switched off through TERT silencing most somatic cells. Since the loss of the protection from telomeric DNA, cells undergo senescence or apoptosis pathway. Conversely, TERT is often upregulated in cells that divide rapidly, including both embryonic stem cells and adult stem cells. In other words, TERT is responsible for elongating the telomeres, which, as a consequence, increases the lifespan of cells by allowing for indefinite division without shortening of telomeres. When the mutation happens in TERT promoter, it results in aberrant expression of TERT. This event allows the chromosome with unstable genome to survive and become cancer cells. From previous work, TERT is aberrant expressed in 90% of aggressive cancer, highlighting this event as a hallmark of tumorigenesis. Accordingly, understand the mechanism of aberrant TERT expression thus represent a crucial outstanding problem in cancer research. Recent research has discovered that the mutation in TERT promoter are the most common genetic alteration in multiple cancer types, revealing a potential mechanism driving increased telomerase activity in tumors. Specifically, one of two positions, G228A and G250A, is commonly mutated in several types of cancer. That is, G228A and G250A mutations may be associated with increased TERT expression. According to this research, it is hypothesis that something can recognize the mutation and somehow activate the aberrant expression of TERT that may contribute to cancer formation. And today's topic will focus on the activation of TERT expression and how GA mutation participate in this regulation. And here now is Qizhang to talk about some findings in this research.
Okay, hello everyone. As previously described, the mutation in TERT resulted in ability of abnormal division in cancer has been already recognized. The G215A or G228A is the most common point alteration in cancer, but the mechanism behind this still lies in unknown. But recently, there might be an answer to this question. Here is the finding from a research group led by Dr. Joseph Costello in University of California at San Francisco. They find out the critical connection between the activation of TERT and the mutation. It might be directed by a transcription factor named GAPA, GABPA, with full form of GA binding protein subunit alpha. Here, in normal cell, without such mutation, the TERT is repressed. Is under repressed and under the strict control of cell cycle, apoptosis, cell arrest, etc. But in a cancer cell harboring the point mutation, the change sequence become a binding site for the gap part and further recruit other transcription factor. By this transcriptional program, the activated promoter gives cancer cell the ability to proliferation aberrantly and contribute to the tumorigenesis. And at the beginning, we don't know the mechanism the mutant sequence initiate the third activation. Since the nucleobase alteration located in the promoter region, it is reasonable to hypothesize that such mutant sequence might recruit transcription factor and hence increase third expressions through transcriptional program and therefore give rise to abnormal proliferation in cancer cell. But how to select a specific transcription factor out of 1,000 possible candidates? And how to know this transcription factor is the right one? It is already known the transcription factor will recognize and bind to specific DNA sequence. Based on that, analysis of mutant sequence could narrow the potential factors to a few family. Therefore, there's a need to confirm whether the G2A mutation is essential for third activation or not. So, let's first repeat the point mutation with different nucleotide. Not only the most common modification in cancer, the G2A, here G228A and here G250A. They also generate the G2C here and the G2T. And then the luciferase assay was performed to test the promoter activity. If the promoter is turned on, it would enable luciferase to synthesize luminance product. And according to the light emission, the promoter is activated or not can be identified. And as the results show here, in the wild type without mutation, the luciferase activity is barely detected. And when the adenine here is replaced with guanine, the expression increased significantly here, this one, and here. And by the way, they also observed that if the sequence was changed into the CCGGATGGG, the third promoter can be partially activated to a health degree of G2A mutation. Okay. And next, after investigation of this sequence through the database, they focus on a specific family of transcription factor named ETS family. And here, ETS family is one of the largest family of transcription factor and unique to animal waste and unique to animal. And there is about 30 member in human. It means there are 30 possible candidates lying here. And once again, by comparison and analysis of the research already done, the number was reduced up to 10. So they aims at least 10 members of ETS family. They generate several knockdown cell models with different ETS family, trying to see lack of which transcription factor could turn down the TERT expression in cancer cell. And here, is a qPCR result, a method to compare mRNA expression relatively, as figure A here shows. Only ETS1 here and ETV3 and GAPA, GABPA, these three factors can lack of these three factors can turn down, can inhibit, repress the TERT expression similarly. So then these three factors was once again be verified through the luciferase experiment as previously described to confirm the knockdown experiment and right side of this page. And this time only the GAPA here, the purple one, and the ETV3 shows the repress ability. But however, in the wild type here, ETV4 hold the repressive ability in the wild type, which rule out the contribution of mutation, indicating the ETV4 repress the TERT by different mechanism, which is independent to the modification of element and was therefore eliminated. Now they have already proved that the GAPA can regulate the TERT activity through previously mentioned knockdown and luciferase acid experiment, but it is still not enough to conclude GAPA regulate the TERT in transcription level. There's still a need direct evidence to prove GAPA would bind to a mutant sequence. So that's why the CHIP sequence was performed. In short, CHIP is a method to analyze the interaction between DNA and protein. The principle of CHIP sequence is to precipitate specific protein with the DNA sequence if binding. Then the protein will be lysed and DNA will be sequenced. So it helps us to clarify the sequence that the transcription factor bind. And here is the result of CHIP sequence. The most of DNA sequence precipitate with GAPA. Here, the most sequence precipitate with GAPA here. Most of them, as you can see here, is the mutant sequence, this and this. And rather than the wild type sequence, consistent with their hypothesis, the mutation might change the sequence into a binding site for certain transcription factor and therefore activate the TERT promoter. And it is also seen in different cancer types, like here, this is a lung cancer or HeLa cell, or here is a breast cancer here. And in contrast, here, the ETS1, another member of ETS family, doesn't grab the mutant sequence. Yeah, there's no peak of mutant sequence. So here comes to the brief summary. In this research, they first confirmed the G2A mutation is essential, not the G2T or G2C. Only the G2A mutation can give rise to TERT activation in cancer. Second, let's select the GABP GAPA out of other ETS family based on the mutant sequence, knockdown and luciferase acid. And more, GAPA can regulate TERT expression. Third, GAPA by chip sequence. The GAPA bind to the mutant sequence specifically. In the conclusion, the GAPA selectively bind and activate the third promoter in cancer.
Hello everyone, I'm Chen Ting Tong. Now I'm going to continue this presentation. According to the previous experiment that both G228A and G250A mutation of the third promoter will enhance the third expression. And the increased expression of third may result from the binding of ETS factor, GAPA, at the mutant sequence. And therefore, the author want to further explore whether the GAPA selectively binds to the mutant sequence in different cancers but not to the wild-type third expression promoter. And in this experiment, the author analyzed the GAPA binding ability with the third promoter in 8 different cell lines and through the GAPA chip sequence. And the cell lines including 6 wild types and 2 cell lines with G228A mutation. And from here we can see that GAPA significantly bind with the third promoter only in G228A mutant cells, including SKNSH, neuroblastoma cells, and HEPG2, hepatocellular carcinoma cells. In contrast, GAPA hardly bind with the third promoter in all of the wild types. In addition, through the peak, we can see that the binding of the GAPA, that most of the GAPA's binding site were close to the G228A mutation, which located at the core promoter, implying that GAPA will selectively bind as the mutant third promoter. And in order to confirm that the finding, the next experiment, the author quantified the binding activity of GAPA with the co-promoter in 7 different cell lines. And 1 is the wild types, and the other 6 are with the G228A or G250A mutation. In this experiment, third minus 5329 will be a control group to compare with the co-promoter in GAPA binding activity. And GAPA chip qPCR is conducted in this experiment. And these results show that GAPA significantly bind at the third co-promoter in whole mutant cell lines. But it almost not bind at the third co-promoter of the cell with the is almost not bind with the wild-type cells, SKMEO28. And therefore, GAPA is mainly bind at the third co-promoter of the cell with the G228A and G250A mutation. And to further support that GAPA can bind at the mutant sequence, the next experiment, the author analyzed the binding activity of GAPA to the mutant sequence through GAPA immunoprecipitation. And the GAPA immunoprecipitated DNA were obtained from the heterozygous mutant cell lines, including HEPG2, SKNSH, and GBN1. And then the DNA were amplified through the PCR, and then transformed into vector and cloned into E. coli. Finally, at least 12 individual bacterial clones were amplified through the PCR and being sequenced. According to the results, input control DNA were inclined to the wild-type area. In contrast, GAPA immunoprecipitated DNA from the mutant cell lines are all significant bias toward the mutant area, represent that GAPA is mainly binding at the mutant sequence. And to sum up these 3 experiments, that the GAPA is selectively identified the G250A and G228A mutant third promoter among different cancers, and therefore regulate the third promoter activity. Next one is Dishu Yang.
Hello, everyone. So the former data tell us the transcription factor GABPA can specifically bind to G228A or G250A TERT mutate promoter and strongly increase the transcription. Here, to realize how this ETS transcription factor bind to DNA in whole genome. So using the genomic-wide analysis of ENCODE SHIP-seq to compare the binding sequence and the binding way of each transcription factors. As you can see, this panel show that only GABPA peaks have strongly binding enrichment scores compared to the other ETS transcription like ETS1 or ELF1. In addition, this panel also show that peaks contain two GABPA motifs have significantly higher binding enrichment scores than just 1 or 0 motifs. It means GABPA may simultaneously bind to two GABPA motifs, and this binding way may be commonly found and effective in whole genome DNA. So we know GABPA may have two motifs. Here, using data analysis software to classify two GABPA motifs peaks into strong peaks and weak peaks, and arrange the motif spacing of these peaks from short distance to long distance. We can find that strong peaks are more likely to have a separation distance shorter than 50 base pair as compared to the weak peaks. However, when the distance is more than 50 base pairs, the strong peaks and the short peaks have no difference. In other words, we can clearly tell the difference and see the intense strong peaks when the two motifs shorter than 50 base pair. Moreover, the increase period seems to occur every 10.5 base pairs. And the interesting thing is the 10.5 base pair distance is really correspond to the major group of the normal bDNA. Highlight the importance of having two GABPA binding sites in the same phase and separate by full helical turns of double-stranded DNA. So two GABPA heterodimer can directly bind each other, forming heterotetramer. So in this picture, we can get two information. First, when the GABPA motifs have a distance shorter than 50 base pairs, strong peaks are significantly higher than weak peaks. So the distance of two heterodimer may be shorter than 50 base pairs. Second, the distribution of the motif spacing are periodically increased every 10.5 base pairs. So GABP can bind to DNA in the same site. So the author, moreover, investigate of the DNA sequence of TERT promoter, which flanks the G228A, G250A mutation site, and less than 50 base pair distance. As you can see, we can find three native ETS binding motifs, ETS195, ETS200, and ETS294. The GABPA will recognize these ETS binding sites, binding motifs or the mutation site. When binding to DNA, GABPB will be recruited to bind GABPA, forming GABP heterodimer, and two GABP heterodimer will bind each other, forming heterotetramer complex. So to determine whether these flanking ETS motifs are required for mutant TERT activation, here performing site direct mutagenesis of each flanking site with or without the G228A or G250A mutation. This panel shows that when G228A or G250A exists, promoter activity is high. But after site mutating ETS195 or ETS200, promoter activity is significantly reduced. However, mutating ETS294 had no effect on promoter activity in the context of G250A. Here. So this data tell us both ETS 195 and ETS 200 are required for aberrant activity of the mutant TERT promoter. In the other hand, the aberrant TERT activation also can be found in a kind of cancer called oligodendroglioma tumor. Although this tumor is wild-type at G228A and G250A, it has 41 base pair tandem duplication away from the native downstream of ETS195. Yes, ETS195 motif in its promoter. This tandem duplication causes two adjacent ETS motifs in the promoter region, and this tandem duplication is really similar to G228A and G250A mutated sequence. Furthermore, mutating either the native ETS195 or the ETS195 site or the de novo junction ETS site can significantly reduce promoter activity. So this data demonstrate that these duplications are required for GABP heterotetramer recruitment. So here we know, no matter G2A, G250A mutation or tandem duplication can cause aberrant TERT activation because of the GABP motifs shorter than 50 base pairs. And when two binding sites exist, they are really important for activation of TERT expression. So the GABP heterotetramer may start to work. And now welcome the Xiaoting to introduce the next experiment.
Continue with the previous person said that flanking EDS motif are required for mutant TERT activation. So next, they want to know whether the EDS motif spacing is essential for mutant TERT promoter activation. And how did they design the experiment? They use site-direct mutagenesis to delete two base pair increments from the native ETS site to G250A mutation, then test each of them TERT promoter activity on these two parts. One have native motif, the other haven't the native motif, ETS200. Because they mutant the ETS200, so it can work. And we can see the first result in TERT promoter activity, which have native motif is more higher than the other. And the second one is we observe clearly periodic behavior during 10 base pair, and it shows the higher peak on these two parts means the most highest TERT promoter activity. And the other field it is because maybe it delete 10 base pair, mean it is delete 1 helical turns. And it let them recruit GABP heterotetramer formation to improve the promoter activity. So through the base pair deletion can change DNA turns, then change GABP dimer or tetramer to influence on TERT promoter activity. And the third result we found that in the wild type data shows the highest promoter activity is at the point which is deletion 6 base pair. So it means the distance from G250A to ETS200 is become 42 base pair, and it's just a perfect 4 helical turns. Reveals G250A and ETS200 just on the same side of DNA. And the same result in the other mutant ETS195 motif. So we can get a summary is GABP bind and switch between both native ETS motif in the context of mutant site, which are essential for robust TERT activation. And the conclusion is, as we can see in this figure, TERT promoter mutation cooperate with
both of these native ETS sites to recruit GABP. Then GABP directly link third promoter mutation to aberrant expression in multiple cancers and overcome replicated sciences and escape apoptosis. Thank you.
Actually, I have a question from the Tsukuba side.
Wait. Which page?
Okay, go ahead.
Okay, so actually, yeah, thank you very much, very clear talk. So then in this case, GABP activate the mutated third promoter, but actually in cancer, the many mutation occur. So that I'm wondering, GABP also activate other promoter, other mutated promoter in other genes.
Excuse me. Could you please repeat your question once again? Sorry. Thank you.
Actually, you show the GAPP activate the third promoter. So my question is, is this possible? Is it possible that GAPP activate the other promoter of other gene in cancer?
I think possible. Of course, it is possible. But here, the most important is they find out the GABPA will bind to the mutation sequence and therefore activate the third promoter and give rise to immortal proliferation ability of cancer cell, and which can be the therapeutic target to maybe cure the cancer in early stage. So maybe, as you mentioned, maybe the activation of GABP can also activate other promoter that also contribute to the cancer chaos. But here we don't know that GABP which promoter my GABP will activate.
Okay, I understand. Thank you very much. Another question from the Tsukuba side.
Thank you for your interesting presentation. I have the first question is the general question. How is the third promoter inactivated in the normal cell? I mean, in which level? Because for inactivation of one gene, there are many levels in chromosome conformation, transcriptional, post-transcriptional, and translation and post-translational. So in which level I want to know, this is the first question. And the second question is, I just confirmed that did the author check the length of telomere of the cell with the mutant promoter? Yeah.
In the first question, in normal cells, when the cells have lost some tumorary genetic suppressor genes like P53, and if one of is P53, and loss of this suppressor gene may let the normal cell can let the normal cell escape from apoptosis and senescence pathway, and these cells and this and these cells, if they mutant in their third promoter and they may proliferate and become cancer cells. So it's a lot of factors combined to make a cancer happen.
So I guess probably his question is about how in normal condition telomere, how the telomere is controlled. So in this data showing the GABP can activate the mutated telomere. So you have more telomere. So the cell will be mortalized, right? They will survive longer. But in normal condition, normal cell, how this out of control of the telomere expression is regulated.
Oh, in normal cells, TERT will be silenced in normal cells. So
By what?
by that's in normal cells.
Any PTM like a phosphorylation, any modification or any translation level,
We don't know, but I think it's in transcription level. Yes.
So in the normal cell, so the telomere expression, the protein is not much.
No, I think it's in normal.
Or it's still expressed, but then not really function well. So I don't.
I'm not pretty sure, but as far as I know, the telomere will forms a specific structure. So the telomerase cannot bind to the Okazaki the lagging strand, so they can keep replicating the telomere, as far as I know. But I believe there is already this mechanism that how the normal cell to regulate the TERT. I believe it is already known, but we need to do more homework. Yeah.
But I guess the question, probably the other thing is, in cancer cell, is the telomere expression high, or is this GFBP transcription factor expression high? So any clinical relevance on either one protein expression? So now you can see the TER expression is abnormal in the cancer, but you also in this paper also show the GABP can.
Maybe it's because the cancer gene have these two mutation, and GABP can bind and activate the TERT promoter, then increase the telomere. This is what TERT. TERT.
I thank you for question for an answer for first question. The second question I asked did you check the length of the telomere in the cell, because some case the gene expressed in transcription level, but in protein level and the function level did not change. So I just want to check because I missed this information in your presentation. I agree with you, because with the mutant promoter, the GABA can bind and activate the gene, but in the protein level, did they lengthen the telomere?
they haven't to test the telomere length.
Thank you.
Yeah, I guess this is a good question. So how this TER upregulator indeed function on the telomere length rescue? Because the tumors, so during the tumor genesis, so the telomere length will be shortening, so they will have the genome instability. But now, certain changing factor can really promote the expression of the TER. So whether the TER indeed to rescue the lens, or it's just doing something else, and so make the cancer cell survival well. So that's a key question: how this TER upregulator involved in the cancer progression or tumor genesis. Right. So any other comments?
Oh, we have a question.
Okay, Kyoto-san, please.
When they are doing knockdown of GAP-PA with siRNA, what is the kind of regulation they're having? Like regulation in terms of mRNA degradation or like the silencing of protein translation?
So can you repeat again the question shortly?
Yes, like when they're using the knockdown of GAP-PA, so what kind of regulation they're having there?
So you knock down the and then so
Have they checked it in the expression level of GAP-PA protein?
so when you knock down the GABP, so you don't have messenger RNA, right? So
Yes.
so what happened? The GABP in regulation of the TER.
In general, the knockdown of GAPA will result in there's no more GAPA mRNA in the cell. So GAPA can be translated into a functional protein. Yeah, so in here, we can see the knockdown of GAPA. No more GAPA transcription factor here. And then third, cannot be expressed. And therefore, here, the third expression will be inhibited. Sorry, do I catch your question? Sorry.
Why I'm asking this question, because there must be in the wild type cell, there must be any regulation of GAP-PA, so that it is like in a, is there any kind of hint in the paper or so in the next level that in the wild type cell, how like GAP-PA must be silenced in the somatic cell when they are in normal condition? Like, is there any kind of this hint in the paper or so?
Okay, so you have a gap, GABP, right?
Yes.
So in the tumor cell, so the third promoter mutation, so GABP bind and upregulate TER expression and leading to the tumor genesis. But in the normal cell, the GABP exists all the time, or then needs some regulatory mechanism to bind some other genes, particularly the third promoter. So what's the regulatory mechanism of the GABP in normal cell in order to avoid loss carbon tumorigenesis or any carbon tumor related gene upregulate?
Okay, in my knowledge, I don't know the mechanism that in the normal cell how to regulate the GAPA to prevent the abnormal activation of TERT. But in other field, the GAPA was believed to get involved in the development of some specific tissue. Yes, but it's beyond my knowledge. Sorry. Yeah.
Okay.
Any other questions? So is there any other questions? Taiwan side and Japan side? No?
We have one more question.
Okay, one more. Okay.
Okay, I'm sorry. Thank you. In this paper, you said that this aberrant activity of TERT promotes cancer, but I want to go to the opposite side of the case, which is the in this case, it's pro-derivative, but I want to talk about the senescence. So I don't know if you are familiar with the term progeria, which is accelerated aging. So I would like to ask about your opinion whether the activity of TERT or telomerase in general contributes to progeria. And also I want to specifically ask about the very interesting periodicity of TERT, because it requires certain distance between the two binding sites to be activated. So whether in progeria, because of the binding sites are out of phase, so that the activity of TERT is silent, and then it leads to cell senescence. Is it possible? Thank you.
So we know the age cell, so the telomere is going to be shortened, right? So they are going to die if they divide more and more. But why the TERT? So what happened to the TERT protein, this enzyme activity or expression during the aging? So why no TERT activity to maintain the distance, the length?
Wait a second. Thank you.
I think so. As you can see, the third, also called the telomerase reverse transcription, is transcriptase is exit in a telomere. So during the age or the cell divisions, the telomere will be short. So I think after aging or cell division, the telomere will cannot to produce the third this protein. So as a cell didn't have the third, they can be live for long term. So,
but the question is why third enzyme is no more exist during in the adult cells, in aged cells, or something wrong on the third during the over time?
I think the expression of third is under really strict control. Maybe in some stem cell will still exist in aged people, but if the telomere in the normal cell is too short to protect its genome in a cell, I think the cell should enter the apoptosis or some cell arrest program to keep the cell can function normally. So I think maybe it's because the stem cells are less and less during our age, so they are less and less can fully function cell in aged people. So sometimes we have to keep the cell that with the least problem. Yeah, and maybe that's why we age.
So I guess probably there's still many studies trying to understand how the third expression or activity is controlled. So there's a many signaling pathway in regulating this kind of the mechanism, but probably still under investigation.
Thank you.
All right. So any questions more? But I guess the time is very limited. It's almost 12:30 in Japan side. So probably we'll stop here for today's lecture, and thank you for the Professor Huang's lecture and also student presentation. And we will see you next week, right? All right, so it's okay. We stop here today.
Yeah, thank you very much, Professor Huang and the Taiwan University members. Thank you very much.
Thank you very much.